All right. So first of all, I'd like to thank our sponsors. We have the consortium partner with SEER, and the consortium major sponsor include Thermo, Bruker, SciEx, Lilly, and other sponsors. So I also want to let you this the second international top-down proteomics symposium, which will be held October 3 to 5th this year in Northwestern University in Chicago. And thanks Neil Kelleher for hosting this symposium. So the early registration deadlines June 1st and abstract due in June 15th. So just right after ASMS. Here are some speakers of this top-down international proteomics symposium. So we're all looking forward to having you join us. So I also want to remind everyone, you're going to ASMS, don't forget the top-down proteomic workshop on Monday evening, presided by Yuri Vanderbilt and Moe Zhou. So there are lots of interesting topics there for you to connect with the top-down consortium. We also have a short course. If you already registered, great, we're going to see you there. It's a 2-day course. There's a lot of topic to be covered on sample preparation, type protein separation, instrumentations, applications, and software analysis. Most critically, we'd like you to join top-down consortium. It's a community, right? It's, we take a village to realize the potential of top-down proteomics, and the membership is free. So welcome you all. Also, the next Proteoform Thursday speaker is Dr. Kelly Sutton from Oklahoma. All right, now finally, it's my great pleasure to introduce Bryon Drown. He's brand new assistant professor from Purdue University. So Bryon actually initially hailed from Massachusetts. So, but he, started to love the life in Midwest. So he received his PhD from University of Illinois in Urbana-Champaign, and received a PhD in 2019. And his thesis work focused on the development of a new antibiotics effective against the gram-negative bacteria and the probes of a very important post-translational modification called ADP ribosylation. So he then become very interested in learning more about top-down proteomics because it's a powerful tool to study post-translational modification. So he joined Neil Kelleher's lab as NCR NISA postdoc fellow. So there he developed the really beautiful methods on top-down proteomics. He joined the chemistry department in Purdue in fall 2022, and he quickly established his own program that used top-down mass spectrometry and organic synthesis to answer challenges in drug discovery. So his title today is Approaches in Targeted Top-Down Proteomics. Here you go, Bryon. The floor is yours. Well, the mic is yours.
Thank you so much, Ying. It really is a pleasure to talk with all of you today. And as Ying said, I'm just getting started here at Purdue University. But I've, for quite a while now, been fascinated by the power of top-down proteomics, and oftentimes in targeted modes. And so I'm actually going to be mostly talking about work that I did in Neil Kelleher's lab, developing more regularized assays for doing targeted TDMS. And so I think before I start talking about it, I really want to mention just how much I appreciate the community of scientists that were in the Kelleher research group, the software developers at Proteomics, and industry partners that Neil has established, because really I learned so much working with those scientists and great individuals, and I'm really indebted to all of them, and all the people on the slide impacted the work that I'm going to talk about today. Right, so we all here care about proteoforms because they encompass multiple levels of variability, and I think we're all very motivated to make measurements of proteoforms because they are a higher value proposition than measurements of genes, transcripts, or proteins alone, right? And so that's because proteoforms are great, often biomarkers for disease. They're good examples in cardiac health. And proteoforms can impact the efficacy of drugs. So there have been quite a few drug campaigns to develop compounds that target the post-translational modification state of oncogenes. So in the case of RAS and tipifarnib, and the targetability of those drugs really, of those targets really depends on the post-translational modification state of those proteins. And we found that, sometimes you can't modify them because the sites of modification aren't even present sometimes. And so I think the method of choice that many of us have chosen is the top-down mass spectrometry approach. So while it may not deliver quite as good of sensitivity and depth of coverage that bottom-up proteomics can deliver, top-down excels at post-translation modification analysis, both the stoichiometry and the combinations of them, and is unique in its ability to identify protein truncations. Of course, we can even perform top-down proteomics on scale in discovery mode. There's this beautiful paper coming that Hoffa published last year on doing this on a remarkable scale in developing the blood proteoform atlas. 22 cell types, many LCMS runs, and finding about 30,000 proteoforms, and that is a remarkable and heroic effort. And we can make discoveries of biomarkers in these unbiased experiments, but oftentimes this experimental design can also be limiting, and it can be really helpful to include an approach that where we look for particular targets. And so when do we need to do enrichment in a top-down workflow? Well, the first reason that we might want to do it is because low copy number. So the number of molecules of proteins in a cell varies across 5 orders of magnitude. And so there are lots of proteins that are low abundance that we're just not going to see in the sea of other more abundant proteins. Or if we have really highly complex proteins that there are going to be many proteoforms that are part of that proteoform family, enrichment can help us identify all of them. And the unfortunate reality is that as proteoforms get bigger, our signal to noise ratio for detecting them goes down. And I think everyone on this call might draw the line differently at what mass things get untenable, but I think we can all appreciate that things get harder as you get bigger. And having an enrichment method certainly can help you deal with large proteins. Okay, so the Kelleher Research Group has developed highly effective top-down mass spectrometry assays that target particular proteins. Some examples that I want to call out initially are the KRAS proteoforms, as well as the apolipoprotein A1 proteins, and the discovery of the beautiful landscape of proteoforms is accomplished by these assays. That being said, I think the reality of these experiments is one that ApoA1 is the most abundant protein in certain lipid particles. So the enrichment isn't exactly particularly challenging. And KRAS, this certainly is a low abundance protein, and it was challenging, but developing this assay took multiple years and multiple postdocs and graduate students. So this was not a turnkey situation. This took a lot of blood, sweat, and tears to get to this point. And so when I joined the Kelleher Research Group, I was fascinated by this question of how do we approach developing these targeted top-down mass spec assays, and how can we make it easier and shorten the amount of time it takes from target identification to acquiring mass spectrometry data. And so when we looked at designing a targeted TDMS experiment, we saw a couple different components that were really important in making an effective assay. And so we need to specifically capture our target by an antibody, most frequently, or some affinity agent, remove non-specific binders from solid support, and then actually liberate our target from oftentimes magnetic beads that we're using. And then finally, clean up and then eventually the mass spectrometry acquisition. And so a lot of factors come into play at each of these steps. So specific capture by an antibody, this is the biggest source of variability. Is every antibody is its own special snowflake, and it's going to behave differently. It's going to have a different epitope. Some of them recognize native epitopes. Some of them recognize denatured epitopes. The clonality, the tolerance for different conjugation methods all vary. Similarly, when you're trying to remove non-specific binders, we even see there's differences in there's no single detergent that works particularly well for removing them. We have surface binding onto plastics, bioproteins that can really limit your ability to detect small quantities of protein. And then when we come to elute beads, again, the antibody has all sorts of issues, right? We've got the clonality of the antibody really affects its ability to be eluted. And then that antibody may actually end up contaminating your sample and get in the way of you detecting proteoforms of similar size. And then when we get to cleanup, getting rid of detergents and then maintaining the solubility of your target are definitely problems that we had. So we felt that there isn't really we thought it was unlikely that we would be able to find a one-size-fits-all approach for any target, and instead recognize a need to rapidly evaluate this landscape of parameters for each target. And so we tried to increase the throughput by which we were developing immunoprecipitations. And so that meant moving to microtiter plates. And so using the Kingfisher Flex is a 96-well compatible magnetic bead manipulator. And by moving to 96-well plates, that allowed us to rapidly screen antibody clones, methods of conjugation, lysis buffers, wash buffers, elution buffers, all on single plates. And then in our initial round of evaluating the performance of immunoprecipitations, we did things on small scale and determined the effectiveness of these IPs by label-free quantification in a bottom-up experiment, and then further validation with Western blotting and silver staining of SDS page gels. Once we had identified a candidate protocol for a given target, that's when we would scale up, because we generally found that we needed a fairly decent amount of input protein for most top-down experiments, and then move to doing either LCMS-MS or doing sample stream tied to I2MS, which is what I'll talk a little bit more about what that is. Anyway, so in this campaign, we developed immunoprecipitation protocols against 41 human targets, or at least attempted to. These targets were selected by by collaborators from the NRTDP as well as HubMap. Involved cell lines and tissues. So some common tissues we were using were lung, kidney, and heart. Generally, we're using 3 to 4 antibodies per target. And so of those 41 targets that we went after, we were able to develop assays that led to detection in the mass spec for 17 of those 41. So again, there's still a high rate of a failure, even when we're exploring a really large landscape of parameters. So I wanted to walk through some simple examples, and then I'll get to some more complex ones. This is an example of P21, of all the different lysis buffers, 2 different antibodies we were using, and this is just one one mode of cross-linking these antibodies to beads. So this is a direct IP. And so from this, we could identify an effective immunoprecipitation, would be shown one on the right here, where from our input, we're completely depleting the lysate of the target, and we are getting some elution from the beads without any contamination from actin in this case. So this would be our binding optimization round, and then we would take these parameters and repeat the process while trying to optimize for elution off of beads. And so we had selected a couple different elution conditions. Most frequently, acidic elution was the most convenient for coupling to mass spectrometry, but sometimes these antibodies, they require, they won't allow elution under acidic conditions. We need to use basic or detergent elution. But in this case, P21 was nice to us and would elute in formic acid. And so we identified some ideal IP conditions for P21 with this antibody clone, lysis conditions, and washing and elution. Okay, so we've isolated P21, and this is a pretty straightforward example where separated still on our more typical analysis mode where we're doing nanoflow LCM-SMS. This is on PLRPS capillaries on a Q-exact HF, and we're able to look at the not super complicated proteoform landscape for P21, but we're able to identify it as well as a couple different mod forms of it. So that's a pretty simple example. We also found that this approach worked pretty well with some, but not all, membrane proteins. And so this is an example of where we were able to immunoprecipitate aquaporin 1 from human kidney tissue. And again, doing our sort of standard LCM-SMS workflow. This is on the Eclipse. I will note that, so we were able to identify a palmitoylated form of aquaporin 1 and isolate the location to this cysteine. One thing I will note that you may catch is that our cleanup method is none. We're actually directly injecting the eluate off of the bead, so that's in formic acid, onto the LCMS, and that's because we found the solubility of membrane proteins really can be quite troublesome in any other cleanup method. And so that leads me to actually a brief discussion about how to clean up samples prior to mass spectrometry. This is something we do quite frequently to remove salts, detergents, whatever other stuff we had to use along the way. And so protein precipitation and solid phase extraction are probably the two modes that were used most frequently in the Kelleher group. Others have used membrane filtration as well as size exclusion chromatography as effective ways prior to MS. I think we found that in the context of an immunoprecipitation, all of these methods had some deficiencies. So solid phase extraction really struggles to get rid of detergents. If anything, I'd say it enriches them. The protein precipitation and membrane filtration, the effectiveness and recovery really drops off dramatically as we reduce the amount of total protein that's going into our sample. And so in immunoprecipitation, where maybe we start with 5 milligrams of total protein, but the immunoprecipitation, after we've eluted, we've got less than a microgram of protein, and finding a protein pellet of less than a microgram is pretty challenging. And membrane filtration, we were getting lots and lots of non-specific binding to those surfaces. So we ultimately found that all of these cleanup methods were unsatisfactory for our workflows, and instead found a technology that was developing that came out of Neil's group, but now is in the sort of the world of commercialization, to be much more effective. And this is the sample stream technology. And this is similar to membrane ultrafiltration in some ways, in that analytes are introduced to a flow cell and are concentrated to a particular region on a membrane. And so while the protein analyte gets focused on the center of the membrane, and small molecules and some detergents that are at concentrations below their critical micellular concentration can elute through the membrane. After washing proteins, then we can reverse the flow and send our analyte either directly to the mass spectrometer in some modes of operation, or we can actually capture that fraction that came out of the flow cell to do other kinds of analysis, oftentimes individual ion mass spectrometry. And so this ultimately ended up being a really effective way for us to clean up samples after immunoprecipitation, and ended up being sort of the standard operating procedure for most of our assays. So I'm going to talk about some more detailed examples, but I wanted to at least touch on some lessons learned that we had from this process. The first is that abundance helps. That when we looked at comparing the abundance of our targets and their that was measured by orthogonal methods and their success rate, there's some relationship between our ability to detect them on the mass spectrometer and how abundant they are. I guess that's not too surprising, but abundance is not all. Is not the only thing that's necessary, right? We've got plenty of examples where highly abundant proteins, we still couldn't enrich them effectively, and we have examples of really low abundance proteins that we could pull out quite effectively. So really, our common fail points. So membrane proteins really still are a real challenge and bear, mostly due to their solubility. Very frequently, we could develop effective capture of proteins, but we could not effectively elute them off of beads. And instead, we needed 1% or higher of SDS. And those cleanup methods that I've been talking about really still struggle to remove that much SDS at well above its critical micellular concentration. And so, potentially other detergents that would be more effective and could help us here. But I think the biggest thing is that, antibody quality really is what makes or breaks the effectiveness of an immunoprecipitation. And we spent a lot of time trying to search for antibodies, match them to targets, as well as match target abundance in different biosamples. And because of all the time that we spent doing that, we actually ended up developing a resource. If you go to target repo.kelleher.northwestern.edu, which brings together human targets, their antibodies, and their abundances, and can help guide the development of these kinds of assays. So those are all the overall lessons learned. I do want to talk a little bit more about two specific targets that I really dived in on their characterization. And these are MEK1 and P53. So I'm going to start talking about MEK1 a little bit in the context of metastatic melanoma. So metastatic melanoma is a disease that is almost uniformly driven by the MAPK signaling pathway. So the MAPK signaling pathway tells a cell to grow, and approximately 50% of patients with metastatic melanoma bear a mutation in the BRAF gene, which sits in the middle of this pathway. And so thankfully, there's the current standard of care is vemurafenib or other similar agents that are selective at inhibiting the mutant form of the BRAF protein. Unfortunately, oftentimes resistance occurs in patients, and so actually, typically now co-treatment with a MEK inhibitor, trametinib, is generally recommended. And so MEK sits in the middle of this pathway. Additionally, MEK is sort of described as the gatekeeper of the MAPK pathway. That's because it's the sole kinase that will phosphorylate ERK1 and 2, and it can actually integrate signals from other similar kinases and can have a really complex control of this signaling. So MEK1 itself has multiple sites of phosphorylation that have been discovered by careful biochemistry and some large-scale phosphoproteomics experiments. The activation segment is in the middle of the sequence. Serine 218 and 222 are recognized as being necessary for catalytic activation of the kinase domain, but we also have sites for feedback regulation. So in the proline-rich domain, as well as the C-terminal domain, you can get phosphorylation by ERK1 and 2, as well as some other pathways can provide some input in tuning its activity by phosphorylation at these sites. So despite having identified all of these phosphorylation sites, almost all assays out in the literature focus on the activation segment, these two sites for phosphorylation. And while these other ones have been identified, the relative occupancy of these phosphorylation sites is unknown. And so we sought to develop measurements of the overall stoichiometry of phosphorylation and try and localize the distribution of phosphorylation across MEK1. So the first step was to develop an effective and specific MEK1 enrichment. And so this was accomplished, again, by immunoprecipitation. On the left here is some Western blotting showing effective enrichment of MEK1, and then a silver stain showing we've got MEK1 here, and a little bit of the antibody that was used to capture it. Thankfully, the antibody in this case actually stays together as the full 150 kilodalton species, and we can actually separate that away. Okay, so the first step of measuring overall extensive phosphorylation can be done with an ensemble measurement. This is, again, using the sample stream flow cell to clean our sample and then directly inject it into, infuse into the mass spectrometer and ionize it. So this is the chronogram of that injection, just over a 3-minute injection and averaging. We can see, here's a distribution. You look at a single charge state, we can see we've got the unmodified singly, doubly, and triply phosphorylated MEK1 in this case. And so I think it's a little bit easier to see this deconvoluted as well as a couple different treatments. So we can see that in untreated A375 cells, that's a model for metastatic melanoma, we have a distribution of 1 to 3 phosphorylations of MEK1. Treatment with different drugs, vemurafenib, trametinib, these are those drugs that hit that pathway. They abrogate the extent of phosphorylation, although there's still some resistant and persistent phosphorylation that remains on MEK1. And then we looked at a couple mutational contexts. So wild type A375 cells, as well as the introduction of 3 mutations that occur in patients that have resistance to BRAF inhibition treatment, and seeing that in some cases, the extent of phosphorylation doesn't change much, but in other cases, we can see a really remarkable increase in the extent of phosphorylation, as is the case for the MEK Q56P example. So using this assay, we found that that the measurements of extent of phosphorylation was rather robust, with really low variance. But I think the first question that anyone ever asks after I show them this data is like, well, where are these phosphorylations? It's one thing for you to tell me that there's an increase or decrease, but where are they, right? And so I think this kind of approach has certainly been done before, where trying to determine locations and occupancies of modification sites by fragment ion relative ratio calculations. This is an experiment in which you isolate the isobaric proteoforms, in this case, for example, doubly phosphorylated MEK1, and then subject it to fragmentation, and then analyze the fragmentation patterns, right? So This has been done for methylation of histones. For example, isolate the triply methylated, and then the fragmentation pattern can tell you where along the sequence of that histone the methylations are. The problem is that when a histone H3, it's a 15 kilodalton protein, and versus MEC1, it's a 43 kilodalton protein. And so when you fragment it and measure those fragment ions by an ensemble method, you're getting overlapping signals. We've got so many theoretical fragment ions that can be produced. They're all sitting on top of each other. In many cases, we also have the E but no D ions that also are overlapping with the most important region of the spectrum. And so you can see some things that are peeking out from the weeds, but ultimately this is not an interpretable spectrum, or at least it's not providing sufficient information. And so we really needed to use an alternative approach to handle this problem of interpreting fragment ions. So this is not an unfamiliar experience for practitioners of top-down mass spectrometry. As things get bigger and more complex, the ensemble spectrum of a lot of species end up resembling the hump of death, is what we described it in the Kelleher group. And this is the same problem where you have overlapping ions and you can't really interpret them. So if you attenuate the ion current down to the point in which they're no longer overlapping, where we get to an individual ion spectrum, you accomplish the problem of we now don't have overlapping, but we now don't really have any features that we can analyze to determine the charge of these ions. And so we're sort of stuck in the same position. So we need to know what these ion charges are in order for this kind of approach to be useful. And so, charge detection mass spectrometry is not new. I can see examples of people doing this on of ions back in the 1960s. They using single image current detectors. But for a long time, this technology required really highly charged, hundreds of charges on those individual ions in order for CDMS to work. These days, the modern charge detection mass spectrometry has improved greatly using electrostatic traps. Now you can you measure the same ion thousands of times in order to get an accurate charge, as well as mass to charge ratio for it. This work largely being driven by Martin Gerald and Evan Williams. But when you evaluate this technology, aside from the fact that it's a fully custom instrument that needs to be fabricated, the lowest charge it can evaluate is 10 electrons. While it does have excellent charge determination, it is very low throughput, having less than 1 ion per second. And they generally have low mass to charge ratio resolving power. And so these are really significant limitations on the ability to analyze fragment ions, where you have a large population, of very, very diverse. And so we thought that this isn't appropriate. And so, a big question that has motivated a lot of work in the Kelleher group for a while has been, can CDMS be accomplished on a commercial instrument? And so I'm going to summarize work that was done by these excellent gentlemen, is basically the implementation of CDMS on an Orbitrap. And so the behavior of individual ions in an Orbitrap mass analyzer is pretty remarkable. These are native proteins, and so the ions actually live a long time in individual ion as you attenuate the signal down, and the integrated signal during the detection event, the slope of that correlates with the charge of that ion. And so that's all well and good with a native ion that is long-lived, but sometimes you're not so lucky, and oftentimes you have individual ions that decay or don't survive the whole transient. And so in this case, a story analysis is necessary. This is where you're actually observing ions through the course of the transient and can see the sites at which an ion decays. And so story analysis really is critical for allowing charge assignment of ions that don't survive the entire scan. And this is definitely necessary for denatured proteins or analysis with an instrument with a high voltage center electrode. And so, but these days, I'd say that we've got the individual ion mass spectrometry workflow worked out pretty well. You can do higher resolution measurements of complex mixtures of proteins and assign charges and construct a mass spectrum. Okay, so what does the fragment ion spectrum look like for MEC1? And so this is that's what I'm showing you here. This is a monophosphorylated MEC, and that I've isolated with a quadrupole, and then subjected it to either electron transfer dissociation or HCD collisional dissociation. And you can see that the distribution of fragment ions is a little bit different for these. We found that, at least for this protein, ETD gave a more, it was an equal opportunity fragmenter, I would say, where we're getting a much more even distribution across the protein sequence. And that gave us a remarkably high sequence coverage compared to HCD, which works well, but there's some preference for fragmentation of particular sequences. And so, yeah, so going forward, we're going to be using ETD on the Orbitrap Eclipse, operating in individual ion acquisition mode. So this also works for highly phosphorylated precursors. So in this case, this is the spectrum of a triply phosphorylated MEC1, isolating with the quadrupole, fragmenting by ETD, and then annotating based on the number of phosphorylations that are present on these fragment ions. And we can see that we can do this because we're able to spread out all these fragment ions across the in the mass domain, have a really high peak capacity, and be more confident in our annotations. Using this approach, we're able to quantify the intensity of the fragment ions at particular sites of cleavage and the number of phosphorylations that are present on them. We can normalize based on, we expect that the fragment ions at an individual site are going to behave relatively similarly to get a sense for where transition points are, or more specifically, where points where we expect phosphorylation to be occurring. So while this is a pretty good map of the occupancy of phosphorylations, there's still a decent amount of variance here, and so we can't effectively measure the occupancy at every single site. So instead, we took an approach where we're averaging across regions between a series of phosphorylation sites that we care about the most. And so that gives us a sense for what's the occupancy of the N terminus, the C terminus, and then these 4 primary sites for phosphorylation. And then so we can eventually map this onto the fractional occupancy at each individual site as a function of the extent of modification of the precursor ion. And you can see that for wild type cells and NRAS K61K cells, there isn't a tremendous difference, but I want to draw your attention to 2 factors. The first is that serine 218 really does not undergo phosphorylation until you have a really heavily high stoichiometry of phosphorylation. It's one of the last sites for phosphorylation, it seems, compared to the C terminus, which seems to have a basal level of phosphorylation. We really only saw phosphorylation of serine 218 in the MEC Q56P, and also suggesting that this mutant, there's some other literature that suggests that it can decouple from feedback regulation. And we can see that it is still getting phosphorylated at the proline enriched domain and the C terminus. So the decoupling of feedback regulation is not lack of modification, but actually maybe a lack of allostery. You can also probably see, based on the size of the error bars, that as we increase in the extent to the highly phosphorylated precursors, this process does break down a little bit. It's not quite as effective. The second area of research was on p53, which is described as the guardian of the genome. So this is a tumor suppressor that in its steady state is polyubiquitinated and degraded, but upon DNA damage becomes activated through relief of that degradation and recruitment at sites of DNA damage. It's also part of the intrinsic apoptosis pathway. It senses that, oh, if I have a small amount of DNA damage, let's repair it. If I have a large amount of damage, let's kill the cell because we have too much genotoxicity. p53 is mutated in 50% of all cancers, and so is of high interest of people in oncology. At the same time, it's a single protein, but it has many functions. Signaling through p53 can give rise to senescence, cell cycle arrest, apoptosis, or as I said, DNA repair, depending on the context. And so how that decision-making is controlled is of high interest. It's also a protein that has many potential proteoforms. And so the number of sites for modification is remarkably high on this protein. And so to the point that with approximately 100 modification sites identified, that in theory gives 10 to the 30 potential proteoforms for p53. However, there are only 10 to the 14 atoms in a cell. So complete coverage of this universe seems unlikely. And so we worked with our collaborators at Harvard Medical School that were interested in the timing of p53 expression dynamics. They see that whether you use ionizing radiation or other DNA damaging agents, you can get different pulsatile natures of the expression of p53. And so the question was, is the proteoform landscape, the post-translational modifications of p53 that matters in decision making, or is it actually some of this expression dynamics that are driving differences? So I set out to develop an immunoprecipitation for p53. I think this is probably the most extensive of the immunoprecipitation optimization trials that we went through. I think we went through 7 different clones under lots and lots of different conditions for doing the immunoprecipitation. This is ultimately where we ended up. And again, this gave us a really clean enrichment of p53. And so that spectrum I showed you earlier of this hump of death for ensemble spectrum, that was p53 after cleanup on sample stream and then direct injection onto a Q-Exacta Plus with a modified center electrode. But when we go to the individual ion spectrum, we can resolve those overlapping species and try and handle the level of complexity that this protein presents us with. And this is an example of the intact p53 mass profile. And so despite seeing 10 to the 30 possible proteoforms, we actually are seeing much fewer. And so one thing that I'll point out as you sort of digest this image is that we are seeing what seems to be a decent amount of phosphorylation events. We could see that intervals of plus 80 Daltons to suggest that post-translational modification via phosphorylation is quite apparent, but there were a number of other mass shifts that we were having trouble identifying. And so comparing across a couple different treatments, whether with Nutlin, X-ray irradiation, ultraviolet irradiation at different time points, we were able to identify a couple of different proteoforms that were changing in their abundance depending on different contexts. In summary, for this section, we developed a robust IP method for p53. I2MS allowed us to access proteoform resolved measurements of p53. The distribution of proteoforms was actually much simpler than we expected, given what bottom-up experimentation told us about the possible sites for modification. And then also we're seeing a relative stability in the proteoform distribution across all those different treatments, which may support the importance of temporal effects. I think with 10 minutes to go, I think I'm going to summarize and say that targeted top-down mass spectrometry methods can be really powerful ways to deliver proteoform landscapes for the proteins that you care about. With knowing the roadmap for how to develop these immunoprecipitations, you can get there. Secondly, the individual ion acquisition methods really extends the capacity and capabilities of the Orbitrap mass analyzer to handle really complex samples, either intact proteins or fragment ions. And it really excels when you need high resolution measurements. And I think that fragment ion analysis is a perfect application of this kind of technology. So this, of course, was the work that was in the context of the Kelleher Research Group. And so I'm so thankful for Neil, all of the my other colleagues who worked with me and supported me and trained me. So thank you to all of you, as well as funding from NIGMS and NCI. As Ing mentioned, I'm just getting going at Purdue University. We're starting out our own research program. We're doing top-down mass spectrometry in the context of drug discovery. And so I'm really excited to get that program off the ground. But thank you all for your attention. And I would be delighted to answer any questions that you may have.
Fantastic talk, Bryon. I could not agree with you more. The targeted top-down is really essential for understanding, biology, and definitely good for drug discovery and also medical applications. So let's open up for discussions. So everyone feel free to just either raise your hand or just turn on your mic and turn your video to ask questions. So if you're too shy to ask, you can type the question in the chat and I can read to Bryon for you. But again, you all are welcome to just, I think you have the ability to turn on your mic and turn on your video to ask questions. Okay, so while we're waiting for more questions to come in, so I will start. I think it's really a great talk. I think summarize all the work in Kelleher's group, and hopefully that really laid the great foundation for your own future programs. So, we definitely share the same passion and with you on targeting, the challenges in top-down proteomics, particularly as the low abundance of the proteins. And many of the top-down study, they focus on the highly abundant proteins and also low molecular weight species, but many of the interesting targets in the biology is actually those low abundant and higher molecular weight species. So I think I like you are did really great job to show up the nice of options available, right, to clean up the samples. So I have a particular question and comments that I like to get started as on the different sample preparation, right, method. And so I know there's all kinds of deficiencies. So what would be your favorite one so far?
Yeah, so it depends on if you have sufficient sample. I mean, precipitation is delivers pretty uniformly in getting rid of particularly detergents. And that's one that we would turn to quite frequently. I think the biggest deficiency with it is that lots of proteins, when they get precipitated, they don't come back. It's a one-way trip.
Yes.
And that's a big problem. And so I know your group has certainly developed ways to try and improve the solubility of those kinds of proteins, right? And so,
right.
Yeah.
I totally agree with you. This is the why people like bottom up because you chop the proteins into peptides, they are much more uniform.
Yeah, you just throw throw 8 molar urea at it, and yeah, it'll dissolve, sure, and then you can digest it, and then everything is lovely.
But it doesn't work for top down. So anyway, so I definitely agree with you that the proteins is once you just precipitate them, it's very difficult to come back. And so I think in our lab, of course, we tried, first of all, is freshly run the samples.
Yeah.
Just making sure your instrument time ready before you analyze, samples. And so also we use azo, photo cleavable detergent, so that avoid which is similar like SDS, but but it does not need the kind of a stringent condition to remove as SDS. So so I think the azo definitely works pretty well so far. And so we now get more questions coming up. So this question from Greg Cap. So great talk. How does effectiveness of I2MS change with size? You mentioned you need 10 charges to see it. Do large protein fragments generally work better? Is there a lower size limit on what I2MS can do?
Yeah, so it's sort of an interesting relationship between you need the ions to survive long enough in the orbit trap in order for you to get good enough charge determination on them. So the really, really big ions, if they're denatured, can experience collisions with gas molecules and dephasing, and then you're not detecting them, or neutral losses, and you can have all sorts of issues happen with big things. At the other end, at the low side, yeah, anything that was charged, like I'd say +6 or less, then we have a hard time. We don't have enough charges on it for the image current produced on the outer electrode to be big enough for us to see it either. And so that's actually why when you look at these graphical fragment maps, the tails are not well occupied. Like we're not getting good coverage of the termini of proteins. But ensemble methods do cover this region very well. And so oftentimes we would do measurements in both modes.
Sounds great. So the next question is from Lauren Adams. Hi, Bryon, nice talk. Do you have any plans to look at the MAC proteoform profiles in patients who have become resistant to treatment? Would be interesting to see if certain proteoform correlated with resistance over time. That's a great question.
Yeah, hi, Lauren. Nice to see you. We worked next to each other in Neil's group. Yeah, I mean that was the plan of the F32. That was the last sub aim was to do that in patient-derived xenografts. We just didn't get there, and I think Neil certainly be interested in this question, but arranging patient samples and getting them, that it's easy to say that you're going to do analysis of patient samples. The hard part is getting them oftentimes.
That's right. And then hard to also get quality samples and how the drug dosage and the durations, so that is definitely important to set up. But glad to see the top-down proteomics has become much more quantitative now.
Yeah.
Okay. So not sure whether there are other questions. Feel free to chat. And so I actually want to raise this important issue that you showed the slides of P53. So you actually showed very nice, proteoforms of P53, and you particularly mentioned that in top-down, they did not see as many as modification as people see in bottom-up. We saw the similar phenomena for troponin I too. So, but in our case, we actually find our, proteoform discovered, and invalidated by a biologist. So it's much more connected to pathophysiology. So I'd like you to address that question: is why there seems to be less post-translational modifications in top-down and bottom-up, and what does this mean?
Well, there's a lot in that question that you're getting to. So I think one thing is, when a post-translational modification site gets added to UniProt, how many observations are actually behind that annotation? Oftentimes it can be really just a single observation that was made. So it's a really low barrier to entry for annotations supported by bottom-up data. I think at the same time, I think the royal we talk about how the importance of stoichiometry and modifications, and I think we do need to be careful when we say, oh, a more abundant proteoform is more important or is the more biologically significant. That's not necessarily true. Like changes at the 1% level or below can have significant biological consequences. And we have to be realistic about what our limits of detection are. And you can see in the case of what really is our limit of detection. And so the noise is really kind of a funny thing with individual ion measurements, because it's like we're either observing the ion or we're not, which is a little different than the way noise works with, say, you're in an ensemble measurement. So that's another whole discussion, but
yeah, no, that's a good point. As I said, I tend to think top down is we observe much more of a consistent, kind of PTMs that is very measurable and quantifiable. So I have to say also there are a lot transient PTMs that, and also different physiological conditions. So I tend to see a lot of bottom-up studies like kind of it's like almost like overexpression. So they actually stressed out they had lots of sites, then probably particularly present in one pathophysiological state. Right?
Yeah.
And of course, what we see is an ensemble of all these proteoforms. When you actually fragment, maybe one site occupancy is very low. That's also possible, right?
So I will say that. So when we go to fragment P53 and try and do that analysis that I did for MEC1, we do have a lot of issues where there are many isobaric species underneath each of these peaks. And so the fragmentation coverage that we do get and we do observe is the region of P53 that is least modified.
Right.
Because all of those isobaric species, they all have fragment ions for the region of the sequence that's not modified. Yeah. And so we get good observations of those.
Yeah.
And there's so much diversity in the other portions that it all gets kind of diluted out.
Right. So the point is not necessarily you're going to observe in your particular state, in your particular sample, that all the sites has been reported in the database, because that's an ensemble of all different conditions. So, so I had to treat those data and knowledge kind of cautiously. Anyway, I don't think we have more questions, and anybody has any last minute question, please raise your hand.
And feel free to reach out to me.
Yes. Yeah. And if not, I think let's thank Bryon again for a really wonderful talk. And, we wish Bryon the great success in his own tenure track. Okay. Thank you, Bryon. Yeah.
Thank you all. All right.
All right. See everybody in ASMS very soon. And yes,